It's safe to say that without a lipid bilayer, a cell is not a cell. In this lecture, I'll cover the metabolism of the primary molecules involved in making up the lipid bilayer of the cell membrane. This will include a discussion of the glycerophospholipids and also the synthesis of the sphingolipids. Now, the glycerophospholipids are, as I said, one of the major components of the cell membrane. You can see how they come about, or how their name comes about, by virtue of the fact that in the middle, as you see here, they have a glycerol backbone. On the right side, they have a phospho component containing phosphate that is very polar. And on the left, they contain the lipid part of the glycerophospholipid, which are two fatty acids attached to the glycerol. Now, this compound is very amphiphilic, meaning that it has a portion of it that's very polar and another portion of it that's nonpolar. Now, this schematic structure of a glycerophospholipid that I show on the screen here, I will carry forward into the next slide. Please note the X at the lower right because this is the point of attachment for other molecules to make the finished glycerophospholipid. Now, you can see on the right that schematic structure, and you can see the X in the box that I've indicated here. If the X is a hydrogen, then the molecule that we're talking about is phosphatidic acid. And phosphatidic acid is not a significant component of the lipid bilayer, but it actually is a precursor for the synthesis of the other molecules below. If X is a serine, as you can see here, then the compound that results is phosphatidylserine. Serine, of course, is an amino acid. If the X is an ethanolamine, we have a phosphatidylethanolamine. If the X is a choline, we have a phosphatidylcholine. And finally, if the X is an inositol, we have a phosphatidylinositol. Now, in contrast to the other additions that were above phosphatidylinositol, the inositol itself does not ionize, but it is a very polar compound thanks to the hydroxyl groups that are on it. Glycerophospholipid synthesis overlaps the synthesis of fats, as we shall see. Now, the starting point for both fats and glycerophospholipids is the molecule you see on the screen, glycerol 3-phosphate. To glycerol 3-phosphate is added a fatty acid in an ester bond at position 1 by the enzyme acyltransferase 1, as you can see. We saw this also in fat synthesis to create lysophosphatidic acid. The lysophosphatidic acid gains a fatty acid on the second carbon of the glycerol, just like fat synthesis, to create the phosphatidic acid that we're already talking about. Now, you can see the phosphatidic acid at the very top of the structure, and that's just mirroring what we brought into this original structure. The synthesis of additional phosphatidyl compounds now can proceed by two different mechanisms, and these are very general in nature, as you will see. Both mechanisms involve the use of what's called an activated intermediate. An activated intermediate is a molecule that has a high energy bond that uses the energy of that bond to donate a part of itself to something else, as we shall see. Now, the two general mechanisms you will see have some parallels, although they sort of flip things around. The first method I will show you is starting with a molecule that is activated through the phosphatidyl compound. Now, this material is called CDP diacylglycerol. The CDP portion of this molecule, of course, comes from the nucleotide CTP, which is used in the synthesis of RNA. And the rest of this is the phosphatidic acid that we had synthesized as of the last image on the screen. So if we take phosphatidic acid and combine it with CTP, we make this molecule. We split out in the process a pyrophosphate. That's the PPI on the side. We started with 4 phosphates, 1 on phosphatidic acid and 3 on CTP. We end up with 4 phosphates, 2 in the CDP diacylglycerol and 2 in the pyrophosphate. Now, the phosphatidyl portion is shown here again for your orientation because that's the part that will be carried forward into the synthesis of the phosphatidyl compound. The CDP splits off not as CDP, but rather as CMP. So it leaves behind one of its phosphates to the phosphatidyl compound. Okay, we can see a reaction here, for example, with inositol. CDP diacylglycerol plus inositol yields phosphatidyl inositol plus the CMP that I said would split off. Now, this reaction requires the energy of that high energy bond of the activated intermediate of CDP diacylglycerol. Thanks to the energy of that bond, the two molecules can be joined together, that is the phosphatidyl compound and the inositol. This reaction does not require energy from ATP because the energy comes from the activated intermediate that was created previously. Now, the second mechanism for the synthesis of the glycerophospholipids sort of flips around the use of the activated intermediate, as we shall see. So, first of all, here's a compound that we want to add to the phosphatidyl part as an X on the figure in the upper right. This is ethanolamine. In order to do this, what we do is we activate ethanolamine using the CDP. Now, before we activated the phosphatidyl compound with the CDP. In order to activate the ethanolamine, we first put a phosphate onto it to make phosphoryl ethanolamine. Now, that phosphoryl ethanolamine is a substrate for making CDP ethanolamine. Now, the way this is occurring is, of course, we have one phosphate on the phosphoryl ethanolamine, we have 3 phosphates on the CTP, and when we combine the two, what we end up with is CDP ethanolamine, and we split out a pyrophosphate, just like we did in the phosphatidyl material. This CDP ethanolamine is also an activated intermediate, so now the energy for creating the joined compound is going to come from this activated intermediate. So we see during the process of this activation that the CMP will split off exactly like we saw the CMP split off of phosphatidic acid earlier. The ethanolamine, meanwhile, will add to what will be diacylglycerol, as we shall see, to make the phosphatidyl compound. So here's the CDP ethanolamine here. Here's diacylglycerol. Now, this is not phosphatidic acid in this case, but rather it's a phosphatidic acid minus the phosphate, because the phosphate here is coming from the activated intermediate. When we put these two together, we make phosphatidyl ethanolamine and CMP. So we've split off the CMP, and once again, we came in with 2 phosphates on the activated intermediate. One of them got carried forward into making the final phosphatidyl ethanolamine. So these 2 mechanisms that I've described, either activating the phosphatidic acid or activating the molecule added to it, are general ways in which glycerophospholipids are synthesized in cells.